All right, let's dive in. Today, we're going to tackle hybridoma technology.
Yeah, definitely a cool one.
It sounds kind of sci-fi, you know?
For sure, but it's real, and it's changing medicine in big ways.
Absolutely. And to help us break it all down, we've got this awesome article from Assay Genie.
It's a fantastic resource.
Okay, so basics first. What exactly are hybridomas? Like, what are we even talking about here?
Okay, imagine combining the power of your immune cells.
The ones that fight disease, right?
Right, exactly. With the ability of, well, let's just say some not so good cells to replicate like crazy.
I'm intrigued.
That's basically a hybridoma. It's a fusion, a mix of an antibody-producing B cell and an immortal myeloma cell.
Whoa, hold on. Myeloma cells, those are cancer cells, aren't they? Is that safe?
That's the genius part. Yes, myeloma cells are cancer cells, but they have this amazing ability to divide endlessly in the lab.
Ah, so they got to provide the engine for this whole thing.
Exactly. By fusing them with those B cells, the ones making antibodies, we create these hybrid cells, hybridomas. They can both produce antibodies and keep making more of themselves. Forever.
So it's a bit of trade-off, a calculated risk.
You could say that. It's like walking a tightrope, but the payoff is huge.
And this isn't exactly brand new science, is it?
Nope, not at all. Back in the 70s, Georges Köhler and César Milstein figured this out.
Those names ring a bell?
Well, they won the Nobel Prize for this discovery.
Wow, Nobel Prize worthy. Okay, now I'm really interested. So how do you actually make these hybridomas? It sounds super complicated.
It all starts with immunization. Basically, we inject a mouse with a specific antigen.
Antigen, like a bad guy molecule that triggers an immune response.
You got it. It's like showing the mouse's immune system a wanted poster.
Okay, so we're training the mouse's immune system to make a specific weapon.
Precisely. This triggers the mouse to make B cells that are like specialists in producing antibodies against that antigen.
And then what happens next?
Then we extract those B cells and fuse them with the myeloma cells in a special lab environment.
So it's like a microscopic matchmaking service.
Uh-huh. That's a good way to put it. Now, this fusion is a delicate process. Not all cells will merge successfully. So scientists have to carefully culture these resulting hybridoma cells. They create a specific environment where only the cells we want, the ones making the target antibody and replicating endlessly, can survive.
So that's not just a simple mix and match. There's a lot of fine-tuning involved.
A ton. It requires real precision to isolate that one perfect hybridoma that's making the exact antibody we need.
We're talking about a single cell here.
Yep. And once you've got it, you clone it. Create a whole army of identical cells all pumping out that same highly specific antibody.
Ah, so that's where monoclonal antibodies come in. These armies of identical antibodies, all thanks to these tiny hybridomas.
You got it.
And I'm guessing this is where things start to get really interesting for medicine, right?
You bet. Monoclonal antibodies have opened up so many possibilities in medicine, in diagnostics, even in understanding how our bodies work at a fundamental level.
Okay, so let's dive into that. What makes these monoclonal antibodies so special? Why is this technology such a game changer?
Okay, imagine being able to diagnose Alzheimer's years before any symptoms even show up.
Seriously?
That's becoming a reality. We now have monoclonal antibodies that can detect those specific protein plaques in the brain way before the disease really takes hold, and that's just one example.
So it's like having a microscopic seek and destroy system, identifying threats before they become a problem.
Exactly. These antibodies are like guided missiles designed to find and bind to a specific target. A protein on a cancer cell, a marker on a virus, you name it.
So we're talking about early detection A&D targeted therapies.
Absolutely. Monoclonal antibodies have revolutionized how we treat so many diseases. In cancer, for example, we have drugs like trastuzumab, which goes after a specific protein on breast cancer cells, and rituximab, which targets lymphoma cells.
So instead of attacking the whole body with chemo, these antibodies are like a special ops team.
That's a great analogy. They go in with surgical precision, which means better treatment and often fewer side effects.
That's amazing. And beyond treating diseases, what else are these antibodies used for?
They've become essential in research, helping scientists study how cells work and understand the complexities of the immune system itself. And they're even playing a key role in developing better vaccines.
Wow, so we've got diagnostics, therapies, research tools, vaccine development, hybridoma technology, and monoclonal antibodies really have left their mark. But with all this amazing stuff, there must be some challenges too, right?
Of course. One big hurdle was figuring out how to stop the human body from rejecting these antibodies, since they're originally derived from mice.
Right, because our bodies would see them as foreign invaders.
Exactly. It's like introducing a new species into a delicate ecosystem. Sometimes it works, sometimes not so much.
So how do you get around that?
Well, that's where things get even more ingenious. We've come up with some pretty clever workarounds like chimeric and humanized antibodies.
Chimeric.
Yeah.
Humanized. Okay, I need more explanation on those. What's the difference?
Think of chimeric antibodies like a blend of mouse and human parts. They have the mouse part that binds to the target, but the rest is replaced with human components.
So it's like giving the mouse antibody a human disguise.
Exactly. So it can slip past our immune defenses.
Pretty sneaky. And what about these humanized antibodies?
Those take it even further. They're almost entirely human with just a tiny bit of mouse antibody left the part that actually grabs the target.
So we've gone from mouse antibodies to hybrids to almost fully human ones. It sounds like there's been a lot of progress in refining this technology.
Absolutely. And it doesn't stop there. We've got these amazing advancements like phage display and recombinant DNA technology. They're giving scientists even more control over designing and fine-tuning antibody therapies.
So it sounds like a very dynamic field, constantly evolving. But before we get ahead of ourselves, I think it's important to acknowledge the foundation, that groundbreaking work that made all of this possible. Hybridoma technology. It
Right.
really is a cornerstone of modern medicine.
No doubt about it. Hybridoma technology has transformed how we understand the immune system, and it's opened up so many possibilities for treating and preventing diseases.
It's a truly inspiring story of scientific ingenuity, but I'm also curious about the challenges researchers are facing today and how they're pushing the boundaries even further. So before we were talking about chimeric and humanized antibodies, those really clever workarounds for the problem of our bodies rejecting those mouse-derived antibodies.
Definitely some elegant solutions there.
Totally. But I'm really curious about how scientists are pushing even further. You mentioned phage display and recombinant DNA technology. Those sound seriously high-tech.
Oh, they definitely are. Phage display is especially fascinating because it uses viruses to our advantage.
Viruses, how does that even work?
Well, specifically, we use these things called bacteriophages. They're viruses that infect bacteria.
Oh, viruses that infect bacteria. How does that help us make better antibodies?
It's kind of like we're borrowing nature's evolutionary playbook. Think of it this way. We create this massive library, billions of different antibody fragments, each one attached to a unique phage.
A phage, like a little virus taxi.
Exactly. And then we expose all these phages to our target antigen, the one we want the antibody to bind to.
So it's like a competition. The phages with the best antibody fragments, the ones that bind really well, they win, right?
It's like survival of the fittest,
Yeah.
but on a microscopic level, we're essentially mimicking evolution in a test tube.
That's mind-blowing. So we're speeding up evolution to find the best antibodies.
Precisely. And the beauty of this is that we can screen tons of different antibody variations at the same time. It's super efficient.
Okay, so once we found the winner, the phage with the best antibody fragment, what happens then?
Well, we can isolate the gene that codes for that antibody fragment and use it to make full-length antibodies.
So it's like phage display finds the blueprint, and then we use that to mass produce the antibody.
Perfect analogy. Now, recombinant DNA technology takes this to a whole new level. This is where we can actually manipulate and engineer antibodies at the genetic level.
Recombinant DNA technology.
Yeah.
I've heard that term before, but can you give me a quick refresher?
It's essentially the ability to cut and paste DNA sequences from different sources, creating these custom combinations with specific properties.
So in the context of antibodies, it means we can tweak and optimize their performance.
Exactly. One really cool example is bispecific antibodies.
Bispecific, so two targets.
You got it. These antibodies have two binding sites instead of one, so they can target two different molecules at the same time.
That's why. What would you even use that for?
It opens up some really exciting possibilities in terms of treatment strategies. Imagine an antibody that can bind to both a cancer cell and a T cell.
A T cell. Those are the immune cells that attack invaders, right?
Exactly. So this bispecific antibody acts like a bridge, bringing the T cell and the cancer cell together.
So the T cell can recognize and destroy the cancer cell more effectively.
You got it. It's like we're directing the immune system's attack force with incredible precision.
That's brilliant.
Yeah.
But I imagine designing an antibody that can bind to two things at once must be super challenging.
Oh, it definitely is. One of the big hurdles is making sure both binding sites work properly and that the antibody stays stable and doesn't bind to anything it's not supposed to.
Sounds like a delicate balancing act.
Yeah.
Are there any other ways scientists are modifying antibodies?
Another amazing development is antibody drug conjugates, or ADCs for short.
ADCs, what are those?
They're essentially antibodies that have a toxin attached to them, like a chemotherapy drug.
So it's like a guided missile with a warhead?
Exactly. The antibody delivers the toxin directly to the cancer cell, minimizing damage to healthy cells.
Wow, that sounds like a game changer for chemotherapy, making it more effective and less toxic.
Absolutely. And we're not just limited to toxins either. We can also use this approach to deliver other therapeutic agents like enzymes or signaling molecules.
So it's like a super targeted delivery system for all sorts of treatments.
Precisely, the possibilities are really endless.
I'm really sensing a theme here. It seems like a lot of these innovations are all about precision and minimizing collateral damage.
You hit the nail on the head. That's a huge driving force in antibody engineering, making these therapies as targeted and powerful as possible while minimizing side effects.
It's amazing how far we've come from those early hybridoma experiments. It really shows the power of scientific curiosity
Yeah.
and our drive to understand and manipulate the natural world.
I couldn't agree more. And as we learn more about the immune system, I'm sure we'll discover even more amazing ways to use its power for good.
It's truly an exciting time to be following this field and seeing how it's shaping the future of medicine. But I'm curious about the challenges that researchers are facing today and how they're pushing the boundaries even further. We've been talking about some pretty incredible advancements in antibody engineering, these bispecific antibodies, antibody-drug conjugates. It's clear that this field is really pushing the limits.
Definitely, it's a fast-moving area with so much potential.
For sure. But what's next? What are the big questions scientists are tackling now, and what could it mean for our health?
Well, one of the most exciting areas right now is figuring out how to use the immune system to fight cancer even more effectively. We're already seeing amazing results with things like checkpoint inhibitors.
Checkpoint inhibitors.
Yeah, they basically take the brakes off the immune system so it can attack cancer cells more aggressively, but antibodies are playing a bigger and bigger role here too.
So it's not just about creating new weapons, it's about helping our bodies fight back more effectively.
Exactly. Researchers are developing antibodies that can block those signals cancer cells use to hide from the immune system. It's like stripping away their invisibility cloak.
Wow, so they can't hide anymore.
Right, and other antibodies are designed to activate specific immune cells, basically sending them on a seek and destroy mission against tumors.
So it's like we're learning how to communicate with the immune system, giving it precise instructions.
That's a great way to put it, but with all this new technology, I have to wonder, are there any risks or downsides to these antibody therapies?
Yeah, I mean, it seems like we're venturing into uncharted territory here.
You're right, there are always potential risks with any new therapy, and antibody therapies are no different. One concern is something called off-target effects.
Off-target effects, what does that mean?
It's when the antibody binds to something other than its intended target, like hitting the wrong button and causing an unintended consequence.
So even with all this precision, things can still go wrong.
That's why testing and monitoring are so important. Before any new therapy is used in people, it goes through rigorous testing in the lab and in animal models. And even after a drug is approved, it's constantly monitored for any problems.
So it's a very careful and thorough process.
Absolutely. Safety is paramount. But another challenge we face is the potential for resistance.
Resistance, like how bacteria become resistant to antibiotics.
Exactly. Cancer cells and viruses can also develop resistance to antibody therapies. It's an ongoing arms race.
So how do researchers stay ahead of the curve?
One strategy is to develop new antibodies that target different parts of the cancer cell or virus, making it harder for them to adapt. Another approach is to combine different therapies attacking the problem from multiple angles.
It sounds like a constant battle, but it's amazing how much progress has been made. It really shows the dedication and ingenuity of these researchers.
I couldn't agree more. It's a truly exciting time to be working in this field, and as we understand the immune system even better, I'm sure we'll see even more incredible breakthroughs.
Well said. Well, it looks like we've reached the end of our deep dive into the world of hybridoma technology and antibody research. We've covered so much ground today.
It's been a fascinating journey exploring this revolutionary field.
We've gone from the history of this groundbreaking technique to those cutting-edge advancements that are really shaping the future of medicine. It's been an incredible journey of discovery.
And I hope our listeners have enjoyed it as much as we have. It's amazing to see how science is constantly pushing the boundaries of what's possible.
Absolutely. And for everyone listening out there, keep asking questions. Stay curious. And who knows, maybe you'll be the one making the next big breakthrough in this exciting field. Until next time, thanks for joining us.